Okay, ladies and gentlemen, so the last thing we're going to talk about in regard to the copying of DNA, which happens during every single cell cycle during the S phase of interphase, is the proofreading ability of DNA polymerase, the enzyme that adds new nucleotides to the new strand of DNA being made. So, as this enzyme, DNA polymerase, adds new nucleotides, there's an error rate of about 1 in every 100,000 base pairs. So every time it pairs an A with a T or a C with a G, every 100,000 times there's typically about one error. It'll pair a G with an A T with a C, things of that nature. But it can proofread as it adds these nucleotides and can actually go back and fix these errors. Now, if a mistake is found, the DNA polymerase will just remove that nucleotide and resume synthesis of the new strand, adding the correct one. A really good analogy is like thinking about hitting the delete button or the backspace button on your keyboard. However, it doesn't always catch the error or the mistake. Because it has these proofreading abilities, the error rate, the mutation rate in DNA is not an error in every 100,000 base pairs. That would be a lot of mutations and a lot of errors all the time. Because we have this proofreading ability, the errors in a completed DNA strand are only about 1 in 10 billion nucleotides. All right, and this is all because the 1 in 100,000 error rate most of the time is fixed, but sometimes it goes unfixed. And that's when we get this 1 in 10 billion nucleotide pairing error rate. So, big takeaway here is that when DNA is being copied, it is also being proofread, so the new strand is as identical to the old strand as possible. So, sometimes you have accidental changes that can occur in DNA that already exists after replication has happened, and the DNA can actually become mutated or damaged as a result. Some of these things include chemicals. You can have radioactivity, such as X-rays, ultraviolet light, molecules, and cigarette smoke. Each cell is going to continuously monitor and repair its genetic material for the reason that DNA can actually be so affected by outside sources of damage. So, when DNA is repaired, and it's the last thing we're going to talk about in regard to DNA replication before we get into how you take DNA, the genotype, and turn it into a physical phenotype. So this is the last thing we're talking about before venturing into that. So we have a pretty simple layout on the left here of how DNA is actually repaired when there is a damaged part. So we have our damaged DNA. We can see the damage here. It's actually going to be cut away by some enzymes called nucleases. You don't need to know what you don't need to know that the enzymes that cut away the damaged DNA are nucleases. The resulting gap is going to be filled with new nucleotides by DNA polymerase. That's this green blob here. That's the enzyme DNA polymerase adding new nucleotides like it does when it's copying DNA. It's doing its same role here. And the other enzymes, ligases, are going to seal the free ends of the new DNA to the old DNA, which we see happening here, kind of like cementing over a hole or something like that, filling a pothole, right, like on the road. So, once this is complete and these two enzymes have done their job, all the DNA has been fixed and hopefully no long-term damage has been done. Okay, so we know that DNA molecules carry instructions for the structure and function of the cell, right? The DNA, the order of those nitrogen bases that exist on those nucleotides, the A's, T's, C's, and G's, within the DNA code. It's the order of those that tells the cell what to do, how to do it, and when to do it. But how are the instructions carried out? How are we going from genotype to phenotype? An even better question is, how does a gene work? We've kind of alluded to that. Excuse me. So let's talk about the genetic code, all right? Because it is just that, it's a code. So the DNA molecule, with its 4 nitrogen bases, is the code for all proteins that are made in the cell. All proteins come from DNA, all right? Remember on the whiteboard I had DNA is made into RNA, is then made into protein. it follows that order every single time. To go from genotype to phenotype, you must go from DNA to protein. Now, genes are made of DNA. We know this from previous units. A gene is the coded DNA instructions. Okay, so a gene is a small segment of DNA that is a certain sequence of nitrogen bases, aka nucleotides, that's going to control the specific production of a protein. You could be an enzyme, it could be a structural protein within the cell, it could even be hemoglobin, which is the oxygen-carrying protein in your blood. It could be any of these proteins. So DNA is inherited by an organism. It dictates the synthesis of certain proteins, and proteins are the link between genotype and phenotype. Proteins are the bridge. Proteins are what get us from our genetic code, which we can't see, to our phenotypes, which we can see. The proteins that are made are going to determine traits that show up in the offspring. Okay. For instance, your skin tone is dictated by certain proteins. Whether you have dark or light complexion, certain proteins dictate that. So how do we define gene expression, right? How do we define expressing a gene as a protein? Well, it's the process by which DNA directs the synthesis of proteins. That's gene expression. You take a gene within DNA, a segment of DNA, and you make a protein from it. That's gene expression, because that protein is going to allow for physical expression of that gene. And the expression of genes is going to include two stages, and they both start with T, and they're both easy to remember. The first stage is transcription, and the second stage is translation, and this is what the rest of this unit is going to talk about. Transcription and translation, which is how you go from genotype to phenotype. It's how you go from DNA to protein. It's how you create a protein in a cell. Okay, so the DNA code is read in a triplet. You can call it like the genetic word. We have a different word for it, but if you look up here, AAT, that is how your cell, how DNA polymerase, how other enzymes in your cells are going to read your DNA in triplets of 3 nucleotides. So we know that proteins are made of building blocks, right? Back from Unit 2, we know the monomers of proteins are correct amino acids. So that means that probably the genetic code DNA, these series of 3 nucleotides in a gene, they have to code for amino acids, right? Because then those amino acids can build proteins. We're getting there. Now there are, we know, 20 different amino acids and 4 different nucleotides. So since there are 20 different amino acids, 4 different nucleotides, there's only 4 different nucleotides because each nucleotide has a different base, nitrogen base. Remember, adenine, thymine, guanine, cytosine. Now, it was discovered that 3 nucleotides in a sequence must specify each amino acid. So this series of 3, this genetic word as we call it, specifies a specific amino acid in the cell, one of the 20. And this would provide for 64 possible combinations of amino acids. So each triplet of nucleotides is called a codon. So instead of calling it a genetic word, a triplet of nucleotides, nucleotide triplet that codes for an amino acid is called a codon. Get it? It kind of sounds like coding. And we'll catch you in the next one, folks.